Okay, so now what we're going to do is we're going to go into our electron transfer chain. This was also in the matrix. Electron transfer chain is in the crista of the mitochondria, which is just another location. But before going in, let's figure what we're bringing into the crista.
Okay.
Okay? So go and add up all the green boxes and tell me what we end up with.
All of the green boxes. So out of like all the ATP that was produced and everything.
Yep, so how many ATP did we get?
So there's, let's see, 4 ATP.
Okay, we have 4 ATP.
Mm-hmm. And then 2 pyruvate.
But we use pyruvate, right?
Oh.
And the next, so all the molecules we kind of use eventually because then we got rid of it.
Uh-huh.
So don't worry about calculating those.
So just look at the ones we ended up with then?
Uh-huh.
Oh, okay.
Yeah.
Okay.
But for this too, like that's important. But the pyruvate, since it was a 3-carbon molecule, but we used it up here to move it in here,
okay.
so we don't really count those.
Okay, so for NADH we have 6, or wait, no. Sorry, I was looking at FADH. My notes are messy. So 10, I think.
Yeah, perfect. 10 NADH come in.
Mm-hmm. And then
How many FADH do we get?
just 2.
Okay. How many CO2?
So do we not count the, oh, never mind.
We basically count everything that we haven't already used.
Mm-hmm.
So since we didn't use this for anything, we'll still count it.
Okay,
Mm-hmm.
so then 6 CO2.
Perfect.
Is that it?
For you. Yeah, that's it. So this is going into the ETC. So I'm sure that'll be a question saying, what are all your products that you have going into the electron transfer chain?
Okay. Do you usually only do recordings if there's just one person?
Normally it's more of a group, just kind of depending on what the schedule's like,
Uh-huh.
or like what other tutors are doing
Uh-huh.
of the day.
Gotcha.
Okay, so we have our, this right here is called my crista, or my cristae. Down here's the matrix, which is where everything else is happening, right?
Mm-hmm.
And up here's my intermembrane. Okay, we have 4 proteins, Roman numeral 1, 2, 3, 4, and then our ATP synthase. Okay, and these are pumps. We have hydrogens. I think this was the good red one. Yeah, it is. So we have hydrogens down here, and we want to shuttle these out from the matrix into the intermembrane. So we want a very high concentration. Times 100 of hydrogens in our intermembrane space. Okay. Because we're putting these against their concentration gradient, right? Because normally you want things to go from high to a low.
Mm-hmm.
Instead, we're going from a low to a high.
So
We need some type of pump.
Uh-huh.
Normally, we always use an ATP pump, right?
Right.
But if we were to use ATP pumps, it would kind of be counterproductive. Instead, we have NADH and FADH, which are electron transporters, electron and hydrogen transporters. So they come in. They came in, they dump all their hydrogens, because you know we just had FAD and then we grab a hydrogen.
Mm-hmm.
So we're able to give our hydrogens here as well as transfer their electrons to these pumps to help fuel the pumps and push the hydrogens out.
So electrons fuel the pumps.
Exactly, instead of ATP. So NADH is going to transfer his electrons, I say as if it's a person, in a different spot than FADH will. So NADH is going to come to this first protein, transfer the electrons, and then the electron is going to jump from the first protein to the third protein to the fourth protein. That's the pathway. First, third, fourth.
So NADH travels first, third.
Mm-hmm. Whereas FADH is going to come in the second, and go second, third, fourth. Kind of a straight line. So you can think of it as NADH doesn't want to invade FADH, so it'll kind of skip over that one and then keep going straight. Whereas this one's just going to input and shoot. Going down.
Okay.
Okay? So we have a key. We want to say per one NADH molecule, we're going to yield 3 ATP.